Hello everyone, myself Swati Wakchoure, Assistant Professor, Department of Pharmacognosy. Today I am going to discuss the topic plant tissue culture. Start from the definition of plant tissue culture. Plant tissue culture is a technique of growing plant cell, tissue, organs, seeds or other plant parts in a sterile environment on a nutrient medium. Plant tissue culture is a technique in which we can use any cell of the plant, any tissue of the plant, any organ of the plant, or any seeds of the plant part in a sterile condition. In a sterile condition means that area are totally free from the microorganism, and where this plant tissue culture is occurred or where it is grow. So these plant tissue culture are basically grow in a nutrient medium. This nutrient medium provide all the essential nutrients for the growth and development for that cell or tissue. And after the sufficient nutrient media, or after the sufficient nutrients, that plant cells are grow into that medium. What is the importance of plant tissue culture? First one, the production of clones of plants that produce particular good flowers, fruits, or have other desirable traits. To quickly produce mature plants, the production of multiples of plants in the absence of seeds or necessary pollinators to produce the seeds means those plant does not having a seeds, so how that plants are grow. So by using the plant tissue culture we can grow that seedless plant in a nutrient medium. The regeneration of whole plant from plant cell that have been genetically modified means we can genetically modify that specific plant species. Then the first importance of this plant tissue culture is that we can produce the clones of plants, and that clones of plant produce the particular good flowers means the any plants which can grow from the plant tissue culture, so that plants having a high quality, that plants having a good flowers, good fruits, or it showing a desirable traits. So by using the plant tissue culture to quickly produce the mature plants. What is the importance of plant tissue culture? The production of plants in a sterile containers reduce the disease transmission means when we use the sterile nutrient medium, or when we grow the any plants in the sterile condition, or in a sterile temperature, or sterile aseptic condition, so that time there is a less chances for the disease transmission. So by using the plant tissue culture, production of plants in a sterile container reduce the disease transformation process. Then next importance is allow production of plant from the seeds the otherwise have very low chances of germinating and growing, that is orchids and the nephthalines. To clean particular plants of viral and other infection, and to quickly multiply these plants as a clean stock for horticulture and the agriculture means we can quickly produce the disease free plants, or we can reduce the infection from the other insects into that plant, because it is totally produce or totally grow in the sterile condition or sterile aseptic condition. The technologies in the plant tissue culture. Explant. So explants means what? When we select the any mother plant, so or when we select the any plant part, then next part is we can select the specific plant part, and into that plant part we cut that plant part into smallest pieces, or that smallest pieces are called as a explant. So these explant are used for the further process of plant tissue culture. Living tissue transferred from a plant to an artificial medium for the culture. So this is very living tissue. Explant is a very small part, or any organ, or any tissue of that specific plant parts, and this living tissue is transferred into the artificial medium for the next culture. Then it can be any portion of the shoot, leaves, roots, flowers, or cell from a plant. So explant is the portion, or we can say that it is a very small parts of the plant part from any parts, that is shoots, from the leaves, from the roots, or from the flowers, etc. So these explant are the living tissue. that can be obtained from the plant part, any plant parts, and that can be transferred into the artificial medium, and from that artificial medium this explant can grow into the whole plant. How this plant tissue culture is occurred? Adult plant cells are totipotent, meaning they have the ability to give rise to fully differentiated plant. Totipotency means what? It is the ability of that plant or ability of that cell that differentiated into the number of cells, and it can fully grow up into the whole plants. So this is the totipotency property. Because of this, it is possible to collect the cell from mature plants and use those cells to produce the clones of that plants. Means we can collect that cell from a mature plant or from the mother plant, and by using this cell we can produce the clones of that plants. What is the basic concepts of plant tissue culture? So first one, modern plant tissue culture is performed under the aseptic condition. So very important part in the plant tissue culture, the aseptic condition is required for the development of the plant tissue culture, or any growing plant in a tissue culture by using the plant tissue culture method. So aseptic condition, sterilized condition means totally free from the organisms. So that conditions, that aseptic condition is totally controlled, means the specific temperature, specific environmental condition, specific climatic condition is controlled into that aseptic condition. So living plants material from the environment are naturally contaminated on their surface with microorganism. So surface sterilization of starting material explants in a chemical solution, usually alcohol and sodium or calcium hydrochloride is required. When we select the mother plant, we can select the any part of the plant, so that time we can cut into the smallest pieces called as a explant. So before starting the culture method, or before transfer into the artificial medium, that explant are sterilized. Why this is important? Because after the sterilization, that explant are totally free from the other dust, other material or other microorganism. So that reason sterilization is very important before transferring into the artificial medium. So which sterilizer is used? Usually for the sterilization of this explant, usually alcohol and sodium or calcium hydrochlorides is required for the sterilization process. Next one, explants are then usually placed on the surface of the solid culture medium, but are sometimes placed directly into a liquid medium when cell suspension culture are desired. So depends on the culture. So when we explants are firstly transferred into the soil culture medium, but sometimes these explant directly transfer into the suspension culture method. So when it is used, when it is desired, then culture media are generally composed of inorganic salt plus a few organic nutrients, vitamins and plant hormones. So the nutrients media, when we transfer the any explant, any cells, any organ of the specific plant, when we transfer into the any culture media called as artificial media, so that artificial media provide all types of nutrient which are required or which is essential to growth and development of that specific plant part. So these culture media contain inorganic salts, it contain organic nutrients, it also contain the vitamins, it also contain the plant hormones. Sometimes we can add the antibiotic to stop the growth of other microorganisms. So this is the content of culture media. Then these are the composition of media for the plant tissue culture. First, salt mixture, organic substances, natural complexes, inert supportive materials and growth regulator. So these are the micronutrient salts. For example, ammonium nitrate, potassium nitrate, calcium chloride, magnesium sulfide, potassium hydrophosphate, ferric and sodium ethylene diamine tetraacetate, boric acid, magnesium sulfate, zinc sulfate, potassium iodide, sodium molybdate, cupric sulfate, cobaltous sulfide. So these are all are the micronutrients. So micronutrient salts, for example, nitrogen, phosphorus, potassium. So what is the role of nitrogen in the nutrient media? So influence the plant growth rate, essential in plant nucleic acid for the DNA, protein chlorophylls, amino acid and hormones. Phosphorus, abundant in meristematic and fast growing tissue, essential in the photosynthesis and respiration process. Potassium are required or necessary for the cell division, meristematic tissue, help in the pathways for the carbohydrate, protein and chlorophyll synthesis. Next one, calcium. Calcium involved in formation of cell wall and root and leaf development, participate in the translocation of sugars, amino acids, ties up oxalic acids. Then iron, involved in respiration process, chlorophyll synthesis and photosynthesis process. Magnesium, it involved in photosynthesis process and respiration system and active in uptake of phosphate and translocation of phosphate and starches. Next one is sulfur. Sulfur is also another micronutrient salt which are required or which are necessary for formation of nodules and chlorophyll synthesis, structural component of amino acids and enzymes. Then manganese. Manganese micronutrients are involved in regulation of enzymes and growth hormones, assist in photosynthesis and respiration. Next one, molybdenum, involved in enzymatic reduction of nitrates to ammonia, assist in conversion of inorganic phosphate to the organic form. Zinc, involved in production of growth hormone and chlorophyll, active in